is not fair on Dr. Mike, is it? So, Mike, have you got the clicker?
The clicker, Ian is...
He's walked off with it. So next up we have Dr. Mike Dineen. So he is an Animal Nutrition and Physiology Senior Research Officer at, I always get this wrong, Teagasc Moorepark in Ireland. Mike's research work focuses on increasing the productive efficiency of pasture-fed lactating dairy cows in an environmentally benign manner. Very nice. He obtained a Bachelor of Science in Animal Science at the University College in Dublin before a Master of Philosophy, is that right, Teagasc? Yep. In collaboration with Queen's University in Belfast. He then completed his PhD at Cornell University under the supervision of Professor Michael van Amburgh, which would be a familiar name to some of you. And his doctoral work focused on describing nutrient supply and animal requirements in pasture-based systems. And he's speaking today on management and nutrition of the grazing dairy cow. So welcome.
Thank you very much. So, yeah, I think, perfect. Yeah, all good. So thanks very much for the introduction, and thanks Nutritech for having me over today. Like Greg, it's first time in New Zealand, so really excited. I'm going to get a few days afterwards now to tour your lovely country. So switching gears slightly, kind of moving away from the acidosis and pH regulation discussion towards nutritional tools that you can use in the field to evaluate things like forage digestibility and rumen function. So for today's talk, I'll try to keep it within time. Everyone's looking forward to lunch. We're going to touch on pasture-based systems, which you're all very familiar with here. We're going to use them as a template of how these new tools can be applied and how we've done it in Ireland over the last few years. We're going to be focusing on new developments in fibre digestibility and thought processes around rumen fill or fill capacity. And then we'll wrap up at the end with a small bit on amino acid requirements and supply of the pasture-fed cow. So again, you're all familiar with pasture-based systems. I like to put up a potential slide here. It was important during the time in Australia to highlight what can be achieved from pasture-based systems. I suppose they lend themselves towards being very high forage inclusion levels. During the summers in Ireland, we can get it down to very low levels of supplementation and 90 to 100% of forage inclusion levels. Those type of diets, as a nutritionist, they're quite scary. They can be high in fibre and very low in starch. And I suppose during my time in the US, it took my colleagues a bit of head scratching to understand how we could get milk from cows on those types of diets. What we do see is that the cows can achieve high dry matter intakes. They can achieve 3.5 to 4% of their body weight. And they can also produce a lot of milk solids on those type of diets. So they can achieve, again, their body weight in milk solids, and we can deliver close to 1,200 kilos of milk solids per hectare on a whole farm basis. And it can also support strong reproductive performance. So I suppose important points to start with. This is just a picture of home. When we were driving over here this morning, I thought I was still in County Cork. So it's very, very similar. I suppose the weather today is helping that as well. We get lots of nice soft days. But I suppose it lends itself towards pasture-based systems, and I suppose converting these human and edible feeds into highly nutritious food, and it's a very important thing that the ruminants perform for us. Our systems are predominantly based in perennial ryegrass, and they're likely to stay that way for the foreseeable future. It has great productivity and persistency, and we're challenged with increasing our output as a country. Since quota abolition, we've increased by 50%. Primarily through an increasing herd size, but with new challenges of today, we have to try to improve productivity on a per cow basis. Some of those challenges, climate change, you're well aware of that here as well. We now have legislation, legally binding legislation that we have to achieve. Water quality is coming on the radar more and more, and also biodiversity loss. So that's driving us towards finding solutions. Some of the potential solutions are optimizing supplementation, so be it the amount of supplementation, the type of supplementation, some of the key nutrients inside in that. Multi-species swards are becoming popular, be it chicory or plantain or even white clover, and also things like fodder beet as well are there. And then there's also the opportunity to genetically improve our plant species or our grasses, so to go after more favorable traits that will help drive performance from that pasture-based diet. So to understand which solution may be the most complementary, Terry, it's important to understand what's coming from our grasses. So what nutrients are they supplying and what might be holding back animal performance. And by understanding the two, we can hopefully increase productivity and nutrient use efficiency from our systems. So I suppose in terms of understanding how to increase performance, during my time in Cornell, I was very fortunate to work with a nutritional model there that's been developed over decades. And when we're using those type of tools, we're always asking the question, what is first limiting our animals? So is it something like physical fill or rumen capacity? Is it metabolizable energy supply, that the cow can't get enough supply in the day? Is it metabolizable protein or even amino acids? And I suppose in terms of nutritional modeling, you're trying to understand which one is reducing performance or holding back performance, remove that barrier, and then reiterate the process. So to help understand what might be limiting, in Ireland, we currently use a fairly standard feed chemistry proximate analysis kind of hybrid. We focus on crude protein, we look at sugars, the typical cell wall components like NDF, ADF, and lignin, and we also look at fat and ash. We then apply a kind of broad organic matter digestibility on all those fractions to give an indication of the quality of that feed. One of the strengths of the CNCPS, the Cornell Net Carbohydrate and Protein System, is its in-depth feed chemistry. So it really digs down deep into that feed as an ingredient and trying to understand how it would behave with the animal. So for example, where we would focus on just crude protein, the feed chemistry in the CNCPS would break that crude protein into 5 different fractions, and each of those fractions have their own behavior or rumen degradation rates and different things, ability to support metabolizable protein. Similar for NDF, there's new chemistry or new methods to break that NDF down further and to gain more information from the fraction of fiber. So by understanding that type of chemistry, it links it with animal factors like rate of passage, and then it predicts how much is likely to break down in the rumen, how much is likely to escape, and also predict things like how much microbial protein is going to be produced from that feed as it interacts with the animal. From that, it can help us understand what might be limiting. Is it metabolizable energy, metabolizable protein, or a specific amino acid? So I just want to go through that feed chemistry around fiber and how it can be useful in a pasture-based system or from a nutrition perspective. So the new fiber analysis is a lab-based analysis. It's in vitro. I suppose it helps in terms of throughput and in terms of labor requirements. So what it does is it takes our plant material and it focuses on the fiber fraction. So in this example, it's ANDFOM, which is your NDF, corrected with amylase, sodium sulfite, and correcting it for ash. So a kind of a more pure NDF fraction. Tony's going to touch a little bit on fiber and the importance around ash correcting your fiber to get a clean and accurate fraction. But what this assay does is it takes that fiber fraction and using long-term fermentation, so after 10 days of fermentation or 240 hours in a vat, it fractionates out the fiber into that which is potentially digestible and that which is totally indigestible. So if it was in the cow's rumen for infinity, it would never break down. By adding in more fermentation time points, so a 30-hour and a 120-hour, it helps us understand how fast that fiber is breaking down, what rate of digestion is occurring, and how much of the fiber is in the different pools. So it ultimately breaks the fiber down into material that's in a fast pool, a slow pool, and an indigestible pool. And we'll just go through a couple of figures here now to see how that helps understand the quality of the fiber. So this is just output from the assay. It's starting at a time point of 0 and goes all the way to 240. And you can see the degradation curve here for an example of maize silage. So maize silage was placed in the flask, and the flask was placed into the water bath. The buffers and minerals and conditions of a rumen were created in that flask, and then rumen microbes were inoculated into it. Over time then, we take the flasks out of the bath and we measure how much of the fiber was left behind. So you can see here the fiber is 100% at the beginning, and then it breaks down over time until it hits its extent of digestion. That degradation curve can be broken apart so we can extract more information from it. Doing some fancy math, putting it on the semi-log scale, we can figure out how much of the fiber is in the different pools. So you can see here represented by the 3 different curves would combine to create the orange curve. So the first part of the curve is the fast pool. You can see here that it breaks down quite rapidly and completely digests. And the rate of digestion of this fast pool for this example was 11% per hour. So 11% of the fiber was breaking down each hour. There was 72% of it in the fiber pool, so a large proportion of the fiber in this feed was in the fast degrading pool. We typically want to have as large of a fast pool as possible. So the bigger the fast pool, it lends itself towards faster ruminal disappearance. It helps with higher intakes. It causes faster eating, we've seen in studies, and also creates more ruminal buoyancy. So the specific gravity of that material, it's more buoyant, and it brings it up into an area where it can be chewed and ruminated. The next pool is the slow pool, or the slowly degrading material. Its rate of digestion is much lower, 2% per hour, and it comprises 20% of the fiber in this example. So we try to avoid the slow and indigestible NDF. So a larger slow and indigestible pool will cause more of a ballast effect, so that physical fill we often hear about in pasture-based systems. It promotes lower intake and also slower eating. So we're trying to avoid that type of material. And then the last pool is the indigestible NDF. So I mentioned it can't be degraded. It's 0% per hour in terms of degradation rate, and it comprises 10% of the fiber in this example. So just to kind of bring those figures together and put it into, I suppose, an animated way, our cows are consuming pasture across the day. There'll be a certain amount of indigestible fiber in that pasture. There'll be a certain pool of slowly degrading material in the pasture, and then there'll be a certain proportion of fast degrading material in the pasture. As that material goes into the rumen, the microbes can attach and start degrading it rapidly, and it'll clear that material from the rumen and help encourage the cow to achieve high dry matter intake across the day. If we present a pasture that is kind of more mature and has a larger amount of the indigestible and slow pool, there'll be less of that fast pool in the rumen, and it won't clear and degrade as quick as the more immature sward, so the space won't be created continuously throughout the day, and will ultimately end up with a lower dry matter intake. So we're trying to maximize the amount of fast pool that degrades quickly. So we wanted to use this new type of analysis from Cornell Group and test how does our pastures behave in that type of analysis. So you can see a picture here from County Cork in Moorepark, and you're all well aware of high quality leafy material. So we'd be aiming for kind of a pre-grazing yield of 2,800 in your numbers, I think. It's around 1,500 in our metrics. And we'd be trying to keep up the proportion of leaf and keep down stem and dead material. So the slides changed a little bit here from switching them over, but this was the example of the output we got when we ran our analysis through the fiber analysis. So we can see here that the rate of degradation of our pastures were extremely fast, and there was a large amount in the fast pool. Here's an example of a more mature grass sample that we would have skipped and preserved as grass silage. But you can see as the maturity increases, the rate of degradation slows down compared to our immature pasture, and the amount of indigestible material is much higher. So the more mature sward had close to 40% indigestible NDF, whereas the immature sward was down around 10% indigestible NDF. We also found through our analysis that because our pastures were so rapid at being degraded, it was important to have an earlier time point in the analysis. So we needed to add in a 12-hour time point to overcome or to gain more information from that analysis. So you can just see the amount of fast pool, slow pool, and indigestible pool, and the numbers here then summarize how much was in each. So our fast pool in our pastures were degrading at 18% per hour, and on an integrated race basis, about 7.5% per hour. So that has all been in vitro, I suppose, in the lab, and I suppose it's important to convert that to the animal. Does it stack up biologically? So to understand how applicable that chemistry was to our pasture-fed cows, we performed an omasal sampling experiment. So to do that, we have a cross-section here of a ruminant. We have a rumen and the reticulum down here, and the orange circle representing a cannula or a fistula. So we took a sampling device and we passed it in through the cannula down into the reticulum, and then we secured it into the reticulo-omasal orifice. So again, the slides are a little off there now, but this weight would go back into the orifice and the opening of the pipe would sit at the reticulomazal orifice where material was leaving the rumen. So we could quantify how much material went into that rumen and how much material was leaving that rumen. So we were able to quantify the fiber intake, which was close to 6 kilos of fiber the cows were consuming in that experiment. And there was 1.6 kilos of fiber flowing out of the rumen. So from the difference, we could calculate how much was digesting. We found that over 70% of the fiber into that rumen was breaking down, and of the potentially digestible, over 80%. So that ruminant had a massive capacity to break down that cell wall material. In North America, in typical indoor-fed diets, that rumen digestion rate would be around 40 to 50% in the rumen, and on a total tract basis, they'd be around 50 to 60%. So just showing when we present that highly digestible material to our ruminant, she can break it down very, very efficiently and achieve high utilization from that cell wall. There's just some pictures of the material that was exiting the reticular rumen. So you can see kind of long vascular strands of lignified material left behind, just showing the capacity of those rumen microbes to strip away that freely available hemicellulose and cellulose when we gave it to her in an immature state. So we wanted to combine the two outcomes then, the predictions from the lab and the observations we had from our animals. So we used the CNCPS to combine the two. And what we found was that the model would predict about 4.3 kilos of fiber would digest in the rumen. So by giving the model the information on the pool sizes and the rates at which they digest and the passage rate of the material from the rumen, the model predicted about 4.3 kilos, or just over 74% of the fiber. In our observations, we found that just over 72% of the fiber digested, or 4.2 kilos. So the model that was developed predominantly on indoor-fed cows and indoor-fed literature was able to predict within 100 kilos how much fiber was breaking down in our pasture-fed cows. So quite encouraging outcomes. We then did a small analysis where we compared if we gave the model the old information, so using lignin times 2.4 to predict indigestibility, and the one time point analysis. We compared that to what the model would predict when we gave it the more in-depth analysis. So you can see the rates of digestion were different between the two, and the predicted milk outcomes was different as well. So with the older information, the model was predicting about 23.5 kilos of milk. And with the new information, the predictions were up to around 25.5. And we compared that to, again, observed outcomes, and it was closer to 26. So by giving that new information to the model and being more specific about our feed chemistry and understanding our feeds, the model could do a better prediction of the outcome or the performance of the animals. Since then, we've gone on to use this type of analysis to understand the effect of things like season. So here's an example of spring, summer, and autumn grass. You can see the differences in the fast pool, the slow pool, and the indigestible pool. So our spring grasses have the lowest amount of UNDF. 10% of the fiber is indigestible in the spring, whereas in our autumn, it was up around 15.5%. And again, that will reflect animal performance. So it's giving us more information to understand what's going on with that cow. You can also see the rates of degradation. So about 10% per hour in the spring versus 7.7 in the autumn. So the rates slowing down. We also experienced a drought in 2018, so quite rare to happen in Ireland, thankfully. But we found that in that kind of droughty, stressed period, our pastures had a much smaller amount of fast pool, so only 50% of the fiber was in the fast pool, and we had a much larger proportion of indigestible material, so 20% of the fiber was indigestible, and our rates of degradation slowed down. So kind of being able to capture what's going on during those adverse weather events. Just using another analysis we did during my time in Cornell, we did a hybrid analysis trial, so kind of using that to show the application of this type of chemistry. So we had 72 corn silage or maize silage hybrids, and they were set in 3 different locations. They were replicated in 3 different locations. So they were all exposed to the same weather conditions, management practices, fertilizer application was all very, very similar. And we just plotted here the outcome from the feed chemistry of those 72 hybrids. So we can find that there was a big range in the amount of fiber in that maize or corn silage, ranging from 30 to 50. So it seems that genetics has a huge impact there in terms of cell wall or cell wall amount. But what was more striking was even at hybrids with very, very similar levels of cell wall, there was a big difference in terms of their UNDF. So NDF wasn't a predictor of the amount of indigestible NDF. So the two of these hybrids would be predicted to have different effects on the animal. So in the analysis, we took 3 different hybrids. We took a high digestible NDF hybrid, represented by the blue line, a control one, so intermediate, and then a slowly digesting hybrid in the green line. We used some rumen capacity metrics that we were after developing over the last number of years. So we took data from Cornell in Italy and work in Ireland, and we've developed kind of rumen max levels, so that how full that rumen can get. And what we're finding is the rumen-fill NDF level is about 1.27% of body weight, whereas the rumen-fill UNDF number is about 0.64% of body weight. So that was set as our maximum fill. The cow couldn't go beyond those max capacities. And then we used the chemistry in the CNCPS to predict how those hybrids would impact performance. So this is just some output from the most recent version of the CNCPS, version 7. And what it does is, over time, it simulates that rumen filling effect. So it uses a number of pools and fluxes. So it accounts for the rate of intake, so how much fiber is coming in over time, how much of that fiber is digesting and disappearing from the rumen, and how much fiber is escaping out through that reticulonasal orifice. And a combination of the 3 determines the rumen pool size. So you can see here for the red line, which was the intermediate one, that was hitting our rumen fill capacity. The green line is the low digestibility hybrid, and that was overfilling the rumen compared to the rumen fill capacity. And then the blue line is the highly digestible hybrid, and that was underfilling the rumen in our simulations. We could do it on a fiber basis and on an indigestible fiber basis. So again, you can see the intermediate one was hitting the rumen fill capacity. The green was overfilling the rumen, and the blue was underfilling the rumen. So there's potential there for that blue one to increase dry matter intake and hit the rumen fill capacity, whereas the green line would have to be down-regulated and dry matter intake reduced. So you can just see some of the model outputs there, and it's just demonstrating that the rumen pool size of NDF in this standard cow was just over 8 kilos. With the low digestible NDF hybrid, it was overfilling the rumen of 8.4 kilos, whereas the high digestibility NDF hybrid was underfilling 7.8 kilos. So there was potential there to increase intake with the high digestibility hybrid. So you can see as we increased dry matter intake in the simulation to meet that rumen fill capacity, the model was predicting increased performance through that upregulation of intake, whereas with the low digestibility hybrid, it was down-regulating dry matter intake and showed a lower performance. So just a kind of way of utilizing the information and the model to try predict how a hybrid might perform in the animals. So again, that's kind of simulation and computer-based. We wanted to use a real-world example to see how this impacts our animals. And this is just some output from our grass-fed cows. So we monitored or used the model to simulate what would be happening in terms of rumen fill. So as the cow is consuming meals, so this is represented here by a meal bout, her rumen starts to fill until she hits a set point where she starts to lie down and ruminate and chew, and the microbes get to work and start processing that material. The black line represents that rumen emptying, so there's disappearance through digestion and a small amount of passage, and the animal goes down to a certain point where she's then stimulated to have another meal bout, represented here by these rectangles. What we're finding in our pasture-based systems is when we've white clover in those swards, we're finding that the rumens are emptying faster, so the rate of digestion and the rate of rumen disappearance is faster, and the animal seems to hit that set point of stimulating a meal earlier, and across the day then she achieves higher meal bouts, and ultimately higher dry matter intake. So that rate of rumen emptying seems to be stimulating dry matter intake. We see it in our studies where we do rumen evacuations. So you can see here a grass-only treatment and a grass clover treatment. And you can see that the grass clover has lost its kind of structural integrity. It's rapidly digesting. It's very, I suppose, the particle sizes are breaking down quicker as well. And just start to see the difference between the two. But again, lending itself towards that rumen disappearance, encouraging dry matter intake. When we look at a systems level, so when we do our larger herd size, those evacuation studies are with a couple of cows, cannulated cows, when we look at our herd level system experiments, we find a consistent increase in dry matter intake. So we see about 0.5 to 0.8 kilos more dry matter intake when we have adequate white clover in our swards. We tend to see it in the summer and the autumn, in the periods where that white clover is representing more of the sward. So you can just see here it's lower in our spring and peaks are kind of late summer into autumn. That also seems to kick on in terms of milk sod performance as well. So we can see in our grass only versus our grass clover, quite similar performance in that early period where white clover is low. And then as we move into the main grazing season, those performances start to separate from the higher clover contents. So just kind of bringing that rumen disappearance all the way through to animal performance. The last place I'd like to touch on is on the protein fraction part of the feed chemistry and the modeling. So within the CNCPS, I mentioned it breaks down the crude protein into 5 different fractions. So we performed this analysis on our pastures, and we found that about 20% was in the non-protein nitrogen pool. So that would represent material that is very unlikely or very unlikely to provide any amino acids to the animal, and it would rapidly break down in the rumen. Over 85% of the material of the protein was soluble in NDF, or in neutral detergent. So again, representing rapid rumen digestion and unlikely to supply feed or UP to that animal. And then only 15% was in the or in the neutral detergent and soluble nitrogen, so in that cell wall matrix. So it's kind of suggesting that a lot of that crude protein would break down in our animal's rumen and not support much amino acid supply in terms of feed amino acids. So again, we wanted to take that to the animal. We used our old-basal sampling experiment to see does that feed chemistry represent what's happening biologically. And we found that the majority of the nitrogen flowing out of our animals' rumens was comprised of microbial nitrogen, and only a small proportion was in the feed and endogenous nitrogen form. So our animals were very dependent on microbial nitrogen or high rumen efficiency in microbial growth. What also was interesting was of that microbial nitrogen, we found that about 77% was in the form of bacteria, whereas 21 to 22% was in the form of protozoa on average. So a very, very interesting outcome in terms of protozoa's contribution to nitrogen supply and amino acid supply. Just summarizing then in terms of, I was discussing nitrogen, we also quantified it in terms of amino acids, and we found that about 1,700 grams of amino acids were coming from microbial protein supply, whereas only just over 300 grams was coming from our feed and endogenous sources. So again, just reiterating how important the microbial protein supply is in these animals. Summarizing it in I suppose, a figure form, as our cows consume pasture, the rumen microbes attach, and they start to degrade that crude protein in the pasture quite rapidly. Those microbes grow and reproduce and create daughter cells, and a certain amount of the pasture escapes fermentation. These two sources then flow out of the reticulomasal orifice to the omasum and onto the small intestine. And in our experiments, we found about 88% of the MP or amino acid supply was coming from the microbes, and only 12% was coming from that feed nitrogen or endogenous nitrogen sources. The CNCPS is just a figure of the inner workings of the CNCPS there. So it's comprised of a number of models and submodels and trying to predict some of these behaviors. So one of those submodels is the protozoal submodel. I mentioned how it was quite striking in our experiments how much protozoa were flowing out of that rumen. So the mechanistic manner of the CNCPS is trying to capture that level of detail and try to really refine the predictions and accuracy of amino acid flow out of the animal's rumens. So just a small touch on protozoa. There's two main subcategories. There's the endodidiomorphs. So this is just a bit of rumen fluid under a light microscope, and it's quite striking of what's going on in that animal's rumen down at the microbial level. And here's an example of a holotrich or isotrich, so very, very different in terms of both protozoa. In the past, protozoa were thought to have been very slow at growing and very inefficient. And Ian mentioned earlier how they can reduce microbial or bacterial growth in the rumen. But I suppose more recently, we're learning how important they are in terms of supplying amino acids to our animals, and also in terms of unsaturated fatty acids, their ability to help with essential fatty acids. So using all that information with the CNCPS, there's new metrics after being developed in kind of building on the likes of the French and the Nordic countries. And they've shown that it's important that we now start to consider protein and energy together, that we can't just focus on MP supply or amino acid supply. We need to connect it with the energy supply as well and help increase our efficiencies of use. So the CNCPS team, like Van Amburgh and his team, and actually Ryan Higgs, who's a New Zealander who drove a lot of this effort, they've come up with new measures in terms of grams of amino acid per mcal of ME and kind of optimal supply targets to try achieve. So they're showing here, for example, lysine, they're aiming for 3.03 grams of digestible lysine per mcal of ME. We use this type of analysis to understand what's being supplied to our pasture-fed cows. And we're finding there may be opportunity to improve the supply of amino acids to our pasture-fed cows, which is quite counterintuitive because we often have a lot of protein in our swards. But what we're finding is that it's extremely digestible, and there may be opportunity to refine those amino acid supplies in our systems. So with that, I'd like just to conclude and try not to hold you from lunch too much longer. There's opportunity to increase performance from our pasture-based systems. In-depth feed chemistry and nutritional modeling are useful tools to help understand how to improve that performance. Immature pasture comprises of a large, potentially digestible pool, which degrades rapidly. New rumen pool size metrics can increase our understanding of rumen fill and function and what might be holding back intake in our animals. And although pasture is high in protein, it's extremely digestible, and there may be opportunity to refine amino acid supplies. So with that, I'd like to thank you for your audience, or for your attention, and welcome any questions.
Thank you very much, and you've certainly caught us up for time. So we have got time for a couple of questions for Mike. Oh, yeah. Christian? I will run this up to you so our webinar audience can hear.
Yeah, thanks for the presentation. That was awesome. I just had a question on the meal bouts that you had there, where you showed that with the digestible diet, there was more meal bouts.
Yeah.
But the meal sizes themselves, do you know if they were bigger or smaller, and did you look at rumen stability at all?
Yeah, so in that simulation, that's a model output. We're using that pattern from studies that have been performed in France and Ireland, and they're finding quite similar meal size, but more meal bouts. From that work. We don't have very in-depth studies on meal bout in Ireland at the moment, but it's an encouraging place to go. And then your second part was?
Just on rumen stability. So thought process is that, I don't know, we've talked about pH not probably be the optimal way to look at it, but mine being that the pH will be more stable because you've got more meal bouts.
Yeah.
So hence hopefully a more stable rumen environment.
Yeah.
So
yeah.
Do you know if they've looked at that at all?
So in that omasal sampling experiment, it's probably not in the same context of that meal bout, but we had rumen pH boluses, and we were tracking the pH over time. And we are seeing a big diurnal effect, so that pH coming up overnight and then dropping down around the first meal, but then quite stable across the rest of the day in areas that we're fairly comfortable with when on a pasture-only diet. So again, a good point, and we must go after that area. Yeah. Yeah.
Great. As I'm walking this microphone over to the back here, really encourage some of the farmers in the audience to write some questions on the post-it notes so that we can take home what it means for us, all of this information and data out on the farm. So, sorry, Sean.
Hey, you were talking about protozoa and how you've been realising that they're more important than we probably thought beforehand. My mind jumped to the use of monensin and the impact of that. Is that possibly not as good an idea as we initially thought?
Yeah, so I suppose very interesting application of monensin. We aren't allowed to use that in Ireland at the moment. So I suppose from these studies, one of the reasons we went after that omasal sampling experiment was the thought process of all that sugar in the rumen from our lush swards and the high liquid turnover. It was really to go and see, do the old thought processes around protozoa stack up. And we found that essentially they have to grow faster than we thought, unless they're able to sequester, but there's rapid rumen turnovers, and it's unlikely that they can. So our findings are showing that they're very, very important in terms of amino acid supply, and they're also been found to consume like chloroplasts and bring some of those essential fatty acids to the small intestine. So I suppose it's a difficult one to answer in terms of we wouldn't have a lot of experience with monensin, but I suppose we shouldn't undervalue those protozoa in their role in the rumen, and Ian alluded to it too in terms of being able to sequester some of those kind of fermentable, fast, non-structural carbohydrates and regulate the rumen environment. So important to consider is all I can really add to your point. Yeah.
Thanks.
Lovely.
I can always rely on Gary for a question. Last question.
Now, I've just got to be careful here. I see all your analysis of bits of fibre and stuff, but some time ago, people actually figured out that if you had a very low dry matter, you could inhibit intake. So if you have ryegrass, like you could have in your typical autumn in Ireland, you might have 12% dry matter on a good day. And I also think that when it's very high moisture, not from rain, but internal moisture, you can limit intake. I just wondered if you had any thoughts on that, because in America it's kind of nice and dry and cut short, and you don't have to eat it very much. But with pasture, you do.
Yeah, so very good point. I suppose all that analysis, while I was in the US doing the chemistry, it was Irish grasses that were coming over across the Atlantic to do the analysis. The French have discovered some of that, and yourselves here in New Zealand, they kind of found a 15% cut point for that reduction in intake when the moisture drops so low, and your point is very important that it was internal water rather than external water. So
Could have been Jean-Louis Perrault who actually discovered that.
Sorry?
Jean-Louis Perrault.
Perrault, yeah, the French and INRA, yeah.
Did a bit together.
Yeah. So
15%.
yeah, so I suppose it comes back to what's first limiting the cow. I mentioned physical fill or ME or MP. Maybe it's something like that at a certain time of the year, that it's low moisture content, or it could be something like time, if we get our time budgets wrong as well. So there's many factors that can limit our animals, and we're trying to understand that in a quantitative manner and release the brakes, I suppose, in these high forage systems and drive performance.
Excellent, thank you very much. So we will head into our lunch break now. And we're returning at 1:15.